Hello, educators, and welcome to GATE. My name is Dr. Adam Wilson from Rush University Medical Center in Chicago. In this video presentation, we'll be presenting on the autonomic nervous system. In this session, we'll compare and contrast the properties of somatic versus autonomic innervation. In addition, we'll also describe the key differences between sympathetic and parasympathetic neurons. We'll talk about a number of structures along the way, including the sympathetic trunks, gray and white rami communicans, and the thoracic splenic nerves. We'll also discuss sympathetic ganglia. Those ganglia, as you'll come to learn, house cell bodies. As such, we'll talk about the pre- and postganglionic sympathetic neurons that are found synapsing at these cell bodies. And toward the end of the presentation, you'll be provided with an example of how the autonomic nervous system is integral to normal sexual and reproductive function. And then lastly, we'll describe a pathology of the autonomic nervous system to you, which is known as Horner syndrome. From a curriculum perspective, it's important to recognize that the autonomic nervous system is often a difficult concept for students to grasp. As such, it's usually useful to start from a very broad perspective and then narrow down to the details. I personally like to take a regional approach to learning the autonomic nervous system so that it's not as overwhelming to students. As a medical educator, I can't stress enough how important it is for students to have a general understanding of the autonomic nervous system. In your anatomy and physiology courses, I do hope that you will give this particular system the attention it deserves. Now let's take a close look at the autonomic nervous system. As we wade into the waters of the autonomic nervous system, I first want to discuss a few basic concepts. The nervous system is divided into both the central and peripheral nervous system. Specifically, the central nervous system contains the brain and the spinal cord. Both of these structures are derived from the neural tube, which develops in the third week of gestation. The peripheral nervous system, however, develops from neural crest cells. Anatomically speaking, the peripheral nervous system is divided into spinal nerves, of which there are 31 pairs, and cranial nerves, which have 12 pairs. Spinal nerves originate from the spinal cord and they exit the vertebral column via the intervertebral foramina. Cranial nerves, however, mostly originate from the brainstem and they exit the cranium through foramina in the floor of the skull. The peripheral nervous system is also divided functionally into somatic and autonomic divisions. Autonomic fibers are further subdivided into parasympathetics and sympathetics. Regarding the somatic system, somatic nerves come in two flavors. The first flavor of somatic neurons that we'll discuss are general somatic afferent fibers, abbreviated GSA. These fibers transmit sensations from the body back to the central nervous system. These sensations can be in the form of exteroceptive fibers, meaning that information is conveyed from the skin about pain, touch, vibration, temperature, and even pressure. Sensations can also come in the form of proprioceptive fibers, meaning that information from muscles, joints, and tendons is conveyed back to the central nervous system concerning joint position and the tension of the tendons and the muscles themselves. Say, for example, that I closed my eyes and I placed my hand behind my head. I know where my hand is positioned in space relative to my other body structures thanks to my proprioceptive fibers. The cell bodies of the general somatic afferent fibers are located here within the dorsal root ganglion. These neurons are considered to be pseudo-unipolar in morphology. That means they have a very long peripheral process that extends from the skin, for example, back to the cell body located within the dorsal root ganglion. They also have a very short central process, extending from the cell body to the neurons housed within the dorsal horn of the spinal cord. Once communication is made with the cell bodies in the dorsal horn of the spinal cord, then the signal is transmitted to the brain for interpretation, Or there can also be interconnections with neighboring interneurons to form a reflex arc within the spinal cord itself. General somatic efferent fibers, on the other hand, are motor neurons. These fibers transmit impulses from the spinal cord to skeletal muscle, which is voluntary. The cell bodies of GSE fibers originate within the ventral horn of the spinal cord. These neurons have a very long axon that is sent out through the ventral roots and through the spinal nerves to eventually reach their target tissues, which in this case would be skeletal muscle. The axons of some of these general somatic efferent fibers can be 3 feet in length or more, especially when you're talking about axons that need to innervate muscles in the sole of the foot. Now that you have an appreciation for the 2 fiber types of the somatic system, let's now look at the visceral system. It is this system that contains the autonomic fibers, divided into sympathetic and parasympathetic fibers. The word viscera means organ, so the fibers discussed here will be transmitting information to and from the organs themselves. General visceral afferent fibers will transmit information such as pain from the internal organs back to the central nervous system. These neurons are considered to be pseudo-unipolar, meaning they have a long peripheral process, a cell body housed within a dorsal root ganglion, and a much shorter central process. And the central process will again communicate with the neurons in the dorsal horn of the spinal cord. General visceral afferent fibers, in addition to conveying information about pain, will also convey information about blood gas levels, blood pressure levels, and vascular distension. The main focus of the remainder of this video will be on the general visceral efferent fibers. These are the motor neurons of the autonomic nervous system, which transmits impulses to smooth muscle, cardiac muscle, and glandular tissue. General visceral efferents are further divided into sympathetic fibers and parasympathetic fibers. As a reminder, the cell bodies of the general somatic efferent fibers were housed in the ventral horn of the spinal cord. In contrast, the cell bodies of the GVE fibers are instead housed in the lateral horn of the spinal cord. These autonomic neurons will send out their axons through the ventral root into the spinal nerves, and they will be destined for their target tissues, which again is made up of smooth muscle, cardiac muscle, as well as glandular tissue. Recognize that somatic motor neurons rely on a single neuron to carry information from the spinal cord to a target tissue. The axons of these neurons could be more than 3 feet in length, especially in the instance of motor fibers innervating the muscles of the sole of the foot. The autonomic nervous system, however, uses a 2-neuron chain system. This series of 2 multipolar neurons is made up of a presynaptic neuron, also referred to as a preganglionic neuron, which has its cell body located in the lateral horn of the spinal cord. It's also made up of a postganglionic or postsynaptic neuron with its cell body located within an autonomic ganglion somewhere outside of the central nervous system. At the synapse between the pre- and postganglionic neurons, both the sympathetic and parasympathetic neurons release the neurotransmitter acetylcholine, which will propagate the signal to the postsynaptic neuron. However, between the postganglionic neuron and the effector tissue, the sympathetic division will release the neurotransmitter norepinephrine, which activates adrenergic receptors. On the other hand, the parasympathetic division continues to release the neurotransmitter acetylcholine in order to either stimulate or inhibit the effector tissue. Now, of course, there is always an exception to the general rule. In this case, the exception is sweat glands. Even though sweat glands are innervated by sympathetic neurons, they are stimulated by the release of acetylcholine instead of the release of norepinephrine. Now let's take a look at the sympathetic division and how it innervates the thorax specifically. So if the cell bodies of the preganglionic sympathetic neurons live within the lateral horn of the spinal cord, then where are the autonomic ganglia located that house the cell bodies of the postsynaptic sympathetic neurons? Well, for the sympathetic division, There are a series of paravertebral ganglia that make up the sympathetic chain, also called the sympathetic trunk, which runs in parallel to the vertebral column. This paired chain is quite long as it extends from the neck all the way down to the sacrum. In addition, synapses can also occur in what are known as prevertebral ganglia. So what's the difference between paravertebral ganglia and prevertebral ganglia? Well, let me remind you that the prefix para means alongside. So the paravertebral ganglia live alongside the vertebral column, whereas the prevertebral ganglia are positioned in front of the vertebral column. Now, personally, I prefer the term preaortic ganglia because it's more specific. These ganglia are located within the abdomen, just in front of the aorta itself. And as you will learn in just a moment, there are nerves referred to as splanchnic nerves that will carry preganglionic sympathetic fibers down to these prevertebral or preaortic ganglia in order for the synapses to occur within these ganglia. In total, there are 22 to 23 pairs of paravertebral ganglia that form the sympathetic chain. There are 3 cervical ganglia in the neck, 12 thoracic ganglia, 4 in the lumbar region, and another 4 to 5 in the sacral region. What is interesting about this information highway is that there are only 14 on-ramps. We'll call these on-ramps white rami communicans. That is, the only way for presynaptic sympathetic fibers to get onto the sympathetic chain is through the white rami communicans that communicate with spinal nerves T1 through L2. Because all sympathetic fibers originate from T1 through L2, the sympathetic division is referred to as the thoracolumbar division, because the fibers originate from the thoracic and lumbar spinal levels. The functions of the sympathetic system that are listed here on this slide to the right are in regards to the organs of the thorax only. As we progress throughout the other body regions, autonomics will come up time and time again. For now, you're only responsible for the autonomics that affect the thorax. Realize that the sympathetic division is the fight and flight division. Picture yourself in a dark alley being chased by a perpetrator. To flee from this perpetrator, what do you need to do? You'll need to increase blood flow to your lower limbs. And to accomplish that, your heart rate and blood pressure will have to increase. The blood flow to the heart will increase so that it can keep up with the demand. And to get more oxygen to your muscles, your breathing rate and depth of respiration will increase as well. In addition, bronchodilation will occur to increase the volume of gas exchange in the lungs. By remembering this simple example, it will help you to remember the key stimulatory effects that sympathetics have on both the heart and the lungs. Before we take a look at the actual pathway these sympathetic fibers take to reach their target tissues, I want to make sure you have a clear understanding of the structures shown here in this image. Let's start by taking a look at the dorsal versus the ventral rootlets. Once the dorsal and ventral rootlets unite, they form their respective roots. So here, the dorsal rootlets are uniting to form a dorsal root. Likewise, when the ventral rootlets converge, they form a ventral root. Sometimes the dorsal and ventral roots are referred to as posterior and anterior roots. Either terminology is fine. Once the dorsal and ventral roots unite, they form a very short spinal nerve. The spinal nerve quickly divides into a larger ventral ramus and a much smaller dorsal ramus. Now let me remind you of the fiber types conveyed through each of these nerves. The dorsal root is considered the sensory limb, if you will, because it only conveys sensory fibers. In other words, it only conveys general somatic afferents and general visceral afferent fibers. Whereas the ventral root would be considered the motor limb because it conveys general somatic efferent fibers as well as general visceral efferent fibers. When the dorsal and ventral roots unite to form the spinal nerve would therefore contain all 4 fiber types, As such, we'll consider the spinal nerve to be a mixed nerve. Likewise, because the dorsal and ventral rami are distal to the spinal nerve, they too would contain all 4 fiber types, so they as well are mixed. As I mentioned a moment ago, there are 14 on-ramps that allow the preganglionic sympathetic fibers to enter the sympathetic chain from levels T1 through L2. The 14 different on-ramps are called the white rami communicans, again entering the chain from ventral rami T1 through L2. They are called white rami communicans because their fibers are myelinated, giving them a white appearance histologically. For the purposes of our analogy, we'll consider the gray rami communicans to be the off-ramps. These communicans are unmyelinated, giving them a gray appearance histologically, hence their name. Let me also mention that all ventral rami of spinal nerves have gray rami communicans connecting them with the sympathetic chain directly, whereas the white rami communicans again are only connected through T1, L2. Once the preganglionic sympathetic fibers have made their way onto the sympathetic chain via those white rami communicans, there are 3 different things that can happen from here. The first option is that a preganglionic neuron can synapse with a postganglionic neuron in a sympathetic ganglion at the same level. Let's take a look at this example. Here is a cell body of a preganglionic sympathetic neuron. It's sending out its axon through the ventral rootlets and ventral root, onto the spinal nerve, and finally onto the white rami communican to enter the sympathetic chain. Once in the sympathetic chain, the synapse occurs at the same level. Here's the synapse, and after the synapse, the postganglionic fiber is sent out through the gray rami communican, and it enters into the ventral ramus. This is the gray rami communican. And this is the ventral ramus. Once in the ventral ramus, the postganglionic fiber is conveyed to the target tissue. The second option is for the preganglionic sympathetic fiber to enter the sympathetic chain and then either ascend or descend to a different ganglion within the chain. Here again is the cell body of the preganglionic sympathetic neuron. The axon is sent out through the ventral rootlets and ventral root onto the spinal nerve and enters the sympathetic chain via the white rami communican. Once within the sympathetic chain, then the preganglionic fiber can either ascend or descend within the chain to another level. In this instance, we're ascending to the level above. At this level above, the synapse occurs within this ganglion, and the postganglionic fiber is sent out through the gray rami communican and enters into the gray ramus. And from there, it again will innervate its target tissue. Here's the third option. In this option, the preganglionic neuron enters the sympathetic chain, but it doesn't synapse at all. Instead, it goes through the paravertebral ganglion and it exits the chain via a back door, which we'll call a splanchnic nerve. Let's take a look at this example. Here again is the cell body of a preganglionic sympathetic fiber that is residing within the lateral horn of the spinal cord. The axon is sent out through the ventral rootlets onto the ventral root, onto the spinal nerve, onto the white rami communican, and once inside the sympathetic chain, no synapse occurs whatsoever. Instead, the preganglionic fiber exits through the back door of the splanchnic nerve in order to make its way to a prevertebral ganglion. Remember, those ganglia were also called preaortic ganglia. Once inside the preaortic ganglia within the abdomen, the synapse occurs, and from there, the postganglionic fiber can be carried to its target tissue. There are a number of different types of splanchnic nerves. The lower thoracic splanchnic nerves specifically are going to emerge from the sympathetic chain and will carry the preganglionic sympathetic fibers to the ganglia located in the abdomen, which again are the preaortic ganglia. The 3 lower thoracic splanchnic nerves are the greater, the lesser, and the least. The greater thoracic splanchnic nerve happens to come from levels T5 through T9, and here's the greater splanchnic nerve shown here on the image. Lower down within the thoracic cavity, you have the lesser thoracic splanchnic nerve, which comes from the levels T10 through T11. And the last splanchnic nerve to arise from the thoracic cavity would be the least splanchnic nerve, which comes from the level of T12. Now let's take a look at the parasympathetic innervation of the thorax. Whatever the effects the sympathetic fibers have on a tissue, parasympathetic fibers tend to have the opposite effect. In the thorax, for example, sympathetic fibers increase heart rate, breathing, and the force of ventricular contractions. Since the parasympathetic system is the rest and digest system, which is an anabolic system that conserves energy, it is largely responsible for bringing tissues back to baseline after sympathetic stimulation. Parasympathetics are also going to regulate the digestive process, including peristalsis and salivary gland secretions. Unlike the sympathetic division, which originated from T1 through L2, the parasympathetic division is referred to as the craniosacral division because it entails cranial nerves and sacral nerves, specifically sacral nerves from S2, S3, and S4. In the head, there are 4 parasympathetic ganglia where the synapses between pre- and postganglionic parasympathetic neurons occurs. For now, don't worry about these, because when we get to the head, you'll learn all about them. However, within the thorax, abdomen, and pelvis, the preganglionic parasympathetic fibers synapse with postganglionic fibers within intrinsic or enteric ganglia, such as Auerbach's or Meissner's plexus. These ganglia are located within the walls of the target tissues themselves, as shown here. This means that preganglionic parasympathetic fibers coming from the brainstem, for example, can be very, very long, whereas the postganglionic parasympathetic fibers are very, very short. Here is a brief summary table outlining the key content we've covered thus far. Recall that the sympathetic division of the autonomic nervous system is the fight or flight division, whereas the parasympathetic division is the rest and digest division. Sympathetics originate from T1 through L2, therefore they are referred to as thoracolumbar, whereas the craniosacral division is another name for the parasympathetic division because it originates from both the brainstem via cranial nerves and the sacral region of the spinal cord from S2 through S4. Next, recall that the length of the fibers differs between sympathetics and parasympathetics. For example, sympathetic neurons have very short preganglionic and very long postganglionic fibers, whereas parasympathetics have really long preganglionic fibers and very, very short postganglionic fibers. And lastly, the location of the ganglia differ between each division. Recall that the sympathetic ganglia are very close to the spinal cord, whether they are pre-aortic or paravertebral. In contrast, the parasympathetic division has its ganglia located in the effector organs, such as the digestive organs, for example. Now let's look at an example of how the autonomic nervous system is integral to normal reproductive function. And this happens to be one example where both divisions of the autonomic nervous system are working in tandem with one another to accomplish a common goal. For the purposes of this example, just remember point and shoot. The P in point represents the parasympathetic innervation of the reproductive organs. Parasympathetics will stimulate an erection. Stimulation activated via the parasympathetics is going to produce nitric oxide, and that nitric oxide is a vasodilator, which dilates the arteries going to the penis and the clitoris. Thereby, engorgement of those vessels causes an erection. And more specifically, it's causing engorgement of the erectile tissues. The S in shoot represents the sympathetic division, which is responsible for causing orgasms, and in the case of males, it's what activates ejaculation. And so together, both parasympathetics and sympathetics are necessary for normal reproductive function. And lastly, I'll present an example of how autonomics can go awry. The symptoms of Horner syndrome are due to an imbalance in the autonomic innervation of the head. In the absence of sympathetic innervation, the parasympathetic division operates unopposed. In patients with Horner syndrome, there is damage somewhere along the cervical sympathetic trunk. And this damage leads to a triad of symptoms, such as miosis, which is pupillary constriction on the affected side, partial ptosis, which is drooping of the upper eyelid on the affected side, and anhidrosis, which is a dryness of the skin on the affected side. Now let's take a look at why each of these symptoms occurs. Let's start with miosis, which is pupillary constriction. Recognize that parasympathetic fibers are responsible for innervating the constrictor pupillae muscle of the eye, whereas sympathetic fibers are responsible for innervating the dilator pupillae muscle of the eye. So with that being said, in Horner syndrome, the sympathetic fibers are out. So the only fibers that are firing and are active are the parasympathetic fibers, causing constriction of the pupillary constrictor muscle. Hence, pupillary constriction results. The next symptom associated with Horner syndrome is partial ptosis, which is a slight drooping of the upper eyelid. Recognize there is a small, smooth muscle referred to as the superior tarsal muscle that helps to keep the eyelid open subconsciously. That muscle is innervated by sympathetic fibers. So if there is a problem with the cervical sympathetic trunk and sympathetics going to the head are out, that means that muscle will no longer be able to contract. As a result, we get a slight drooping of the upper eyelid on the affected side. And the last symptom is anhidrosis, which is a dryness of skin and an inability to sweat on the affected side. Do recognize that sweat glands are innervated by sympathetic neurons. Therefore, with no innervation coming from sympathetics on the affected side, we have an inability to sweat on that side. In addition, also recall that sympathetics are responsible for vasoconstriction of cutaneous blood vessels. With the sympathetics out, that means the blood vessels become vasodilated. As such, it can cause a red or flushed face on the affected side. This is a great example of how both sympathetics and parasympathetics complement one another to maintain normal function. If you don't already use this example in class, I hope you'll consider using it next time. Thanks so much for listening to this introductory video on the autonomic nervous system. The GATE instructors look forward to meeting you. We'll see you soon. Take care.